You were so like.
So are you going to let me infringe on your time a little bit since you're all here and ready to go?
As long as you let us out.
Give me two. Oh. Deals. Well, this is a new experience for me. Several of you I've not had in class before, and that's typically, I've had everybody in class because they've taken cell biology, but I was on sabbatical when many of you took the cell biology class, so it'll be a learning experience for you and me. Syllabus is pretty self-explanatory. Please look it over, and then I have you sign it at the end. Basically, this is our contract about how the course works. It's set up the way cell biology was set up. If you haven't had that class, that doesn't mean anything to you. Probably the biggest thing that is a little confusing to students that haven't had me before is the optional point system. I think Morton McCombs uses optional points as well. But it's a way to gain some extra traction in the course. But the big difference is that it's not the same as bonus. When you're calculating grades with bonus, you only add it to the numerator. But in optional points, you add it to the numerator and denominator, so it doesn't have quite the impact that just bonus points do. But it's a way for you to do some different exercises and experiences that should not only help you for the exams, but also help you to get a better course grade. There is a cap of 4%, and so the maximum you can raise your grade is 4%. But 4% can be fairly significant at the end of the semester. I mean, very much oftentimes the difference between a letter grade and a plus, or a minus and a whole letter grade. So I would urge you to take advantage. One way to rack up a fair number of optional points later in the semester will be to do an exercise called ID Factor. And it's a good way to learn some things you need to learn to do your unknown for the semester. It's also a good way to prepare for the second lab exam. So look at the website. You all know where the course web pages are for science courses. I think it's mainly Rhodes and I that use course web pages. I don't use Moodle. We use a website. It's now linked to the college's web page, and so if you go to just Franklin College and go to academics and then majors and choose biology, then there's all kinds of different things on the left-hand side. One of them I think is course information, and there are the course web pages, including microbiology. So check that out if you have not seen that. There are PowerPoints there. I've got all the PowerPoints for the course already posted there. There are study guides. There are old exams. And then I've also posted some of my past old lecture notes. And they change from semester to semester, but the skeleton is usually pretty similar. And so if you like to look at those, particularly after you've looked at your lecture notes, and it can help you to fill in some gaps. And so those are all posted there as well. If I can remember, I will also tape lectures, and then Natalie just posts those on the website as well. So if you're going through your notes and you're missing something and you want to go back and hear the lecture, you can. Or if you've missed a class for some reason, you can pick up that lecture, but I'll have her post those. If you like to use those, and I'm falling behind in terms of remembering to turn on my recorder every day, you might just remind me and say, have you turned on your tape recorder today? But it's just an easy way to kind of have some backup information for you. So any questions about the syllabus? Class meets 3 lecture periods per week, and so that is the Monday, Tuesday, and Thursday at 9 o'clock. Wednesday is lab from 9 to 10:50. How many of you have an 11 o'clock class? Okay. You need to just kind of watch out then and make sure you get out of here in time for your 11 o'clock class. And then Friday at 9 is also a lab period. Okay, that's a 1-hour period. And sometimes we will be finishing something up from the Wednesday lab. Sometimes it will just be a short lab exercise. When we do microscopy, for example, there's a couple of short staining procedures that you can get done in an hour. And so all of those are posted on the syllabus by day. Please make sure that you have read the lab handout for the particular lab prior to coming to lab. Okay? How much are they selling the lab packet for now, do you know? 10 something?
Yeah.
Okay, that's a real bargain. Using the same book we used last year, so if you know someone who has an older copy of the book, that's fine. Even if it's probably a couple of editions old, that's probably not a huge thing. I like to use the book for context. If you can get in the habit of reading chapters before we talk about it, and all the reading assignments are on the syllabus, it's going to be to your benefit to do that. That'll give you some kind of overview of what we're talking about. There's some things in the chapters that they cover that I don't cover. There's some things, a lot of things that I cover in class that isn't covered in the book. So you can use the book as kind of a complement to your notes. Exams are note-based. I don't pull things out of the book that we haven't covered in class. And so really the emphasis should be on your lecture notes. And the study guides really help you to, if you're disciplined, go through those lecture notes on a regular basis. Study guides oftentimes end up being exam questions. And so if you can get in the habit of doing study guides, I would really encourage it. If you would like to come in and go over answers to study guide questions, I'll be glad to do that for you. If you want to work in groups, that's fine, but it's probably not a great idea to just split up the study guides and have John do 1 through 5 and Ariel do 6 through 7. And you really should do your own study guide questions, but then if you want to talk through your answers with one another and see if you're coming up with similar answers. But typically they do follow the lecture notes pretty closely. So if you can get in the habit of looking over your lecture notes after class, sit down with the study guides and start writing out answers. You're studying for the exams, okay? So if you can do that, you're going to be studying for the exams on a routine basis, and then you're not going to be trying to cram all this stuff in a night or two before. So it's the same thing that probably you've been told in all your other classes, but there's some tools on the website to really help you out. Old exams can be useful, particularly since many of you haven't had an exam from you before. So you can see what the length of the exams are, the kind of questions that I ask. Sometimes the same questions pop up on an old exam that are on your exams, but I tend to sit down and write new exams every semester. So they're there for not memorization purposes. Don't study by just memorizing an old exam, but use those to get a sense of kind of what the exams are like. I do typically about half objective questions, multiple choice. I do agree and disagree questions. I do 8 to 10 point essays. So the format of the exams that are on the web are pretty indicative of what you'll see. We can talk about that more as the first exam comes up. So, any questions for me so far? Well, I'm going to torture you, and we're going to start today. Okay. One of the things that educational research says is that students are much more likely to be engaged in a class if they believe that the information is relevant. Makes sense. Okay. So I guess the first thing I would like to do for you is to argue that this is a very relevant course. Probably the thing that I can say to you that would hit that home the most is that what would happen if you could magically be a grand wizard and say all single-celled organisms are gone? What would be the consequences of that? We eliminate all microbial life.
Everything would die.
Everything would die. Okay? Life cannot exist without microorganisms. And today we'll talk a little bit about that in the context of why should we study microbiology. Okay? But life cannot exist without microorganisms. Mostly the general public has a misconception about microorganisms, concentrating very much on the detrimental activities of microorganisms. There are some detrimental things that they do. Obviously, human disease is a big one. And so many people feel like, well, if we could just get rid of these bugs, these critters, life would be rosy. No, there would not be any life. Okay. Second relevance is, in areas related to microbiology, there are a lot of jobs and careers in science. Okay, so if you're worried about a career when you graduate, and I know you all are, then microbiology can have a lot of relevance. Areas of biotechnology depend upon microorganisms to do a lot of the work to manufacture a lot of the biotech products. Pop a gene into E. coli and let E. coli produce the human insulin rather than trying to harvest it from the pancreas of an animal. Okay? Genetics. Much of molecular genetics was figured out in microbial models. What we know about, for example, DNA replication, protein synthesis, was all figured out pretty much in E. coli first. And then once we had that simpler model figured out, then we could kind of extend that knowledge to more complex eukaryotic systems. So microorganisms are kind of the lab rat of molecular biology and biochemistry. Huge area that we're going to spend a fair amount of time talking about in this class is immunology. What is the human immune response to microbial infection? Other areas, if you are interested in ecology, for example, microbial ecology is a huge area of study. If you are interested in medicine and disease, epidemiology, the study of the causes and distributions of disease, is a very important part of public health study. So microbiology kind of has its fingers all over the scientific world. And so if you're looking for jobs, having some of these skills and applications can be really important. For example, John wants to go to a medtech program. He needs to know micro because you need to know micro to work in the lab, but you also need to know immunology, particularly to do a lot of the antibody-based testing that's really becoming a big part of microbiology. David, you're interested in dentistry, right? Microorganisms, huge responsibility in dentistry. Periodontal disease, tooth decay, etc. Several of you are interested in medicine. Obviously, all of the microbial and viral diseases are very relevant to your area of study. So there should be a lot of things to keep you engaged. I will occasionally send out articles for you. I probably won't test you on those, but if I find something interesting, I just like to share it with you. And then if you find that you'd like to do a little bit more research into that, it might be possible for you to do some optional point work. Okay, so sometimes those kind of articles just kind of get a thread going and get you going on a little project. If you run across things that are interesting, send them to me. I'll be glad to share them with the class. But I think that if you can kind of find topical information about the area you're studying, it's just a lot easier to get engaged in it. And if you're more engaged in it, you're probably going to keep up and study more. So that's going to be important. So let's start out with what? What's going to be the province of this course? What are we going to be studying? What are our subjects of study going to be? Pardon me, Hannah?
Microorganisms.
Okay, be a little bit more specific. Give me an example of a type of microorganism. Bacteria. Okay. So when we say microbiology, obviously we're talking about studying small things, right? So microscopy is going to be something that's going to be incredibly important, particularly in the lab part of this, of course. Okay. Well, bacteria are microscopic, and we'll spend a lot of time studying bacteria. As a matter of fact, that's really going to be the primary emphasis in terms of the type of organism that we're going to be studying. So in some respects, this course probably should be titled instead bacteriology/immunology than microbiology. Microbiology is kind of a generic term that can describe a lot of different kinds of microbial entities. What's some other possible fair game for a microcourse besides bacteria? Virus. We will talk a little bit about virus, but primarily in the context of human health and the immune response to viral infections. If we have a larger department, we could spend easily a whole semester having an interesting virology course. okay? But we've got to kind of pick and choose our battles. So we will talk about virus, but in some very limited context. Anything else that might typically be the province of a microbiology course, besides these two?
Biodegradation. Organisms do it anyway.
Okay, and many of those are bacteria, but some of those are fungi. In some microbiology courses, fungi are a big part of a microbiology course. I'm really not going to cover fungi because we let Huygens do that in botany. Okay, what were you going to say, Nicole?
Antibodies?
Okay, antibodies definitely is part of immunology. Let's kind of talk about organisms now as opposed to molecules. Okay. Another entity that sometimes is covered in a microbiology course are protozoans. There are a number of protozoan diseases. Okay. But we're not really going to talk about protozoans much at all. You're going to have to get your exposure to protozoans and a little bit of stuff about protozoan diseases. Some of you, does any of you have inverted zoology? Was Dr. O'Neill talking about some of the protozoan diseases in zoology? Malaria? No? Okay. So depending upon where you are and what the kind of overall curriculum of the program is, microbiology courses can cover a fairly large range of organisms. But as I said, we're primarily going to be a bacteriology course with some heavy emphasis on immunology. The reason we're going to include so much immunology is twofold. Number one, it's very important to understand some basics of immunology. But number two, because of our affiliation with the MedTech programs, we have to have a significant component of immunology in our microbiology course because we don't have a separate immunology course. Maybe sometime down the road we will. And if that's the case, then maybe this course will evolve into something more that includes virus, for example. We're kind of dictated by various affiliations that we have. So, how are these organisms related? One of the things we're going to spend some time talking about in this course, particularly at the beginning, is taxonomy. Way back when Linnaeus developed the binomial tax classification system, genus, species, binomial, two names, there were only two groups of organisms according to Linnaeus. What were they? Everything was either one of two types of organisms. That was after Linnaeus, okay? We didn't know about prokaryotes in the 1750s. Animals and plants, okay? The two groups of organisms, according to the original Linnaean taxonomy, were plants and animals. So where did bacteria fit in the Linnaean system of classification? Anybody know? 50-50 shot. Twice. Have you heard the term flora and fauna before? Flora referring to plants, fauna referring to animals. Well, one of the things that we will talk about a little bit even today is the concept of your bacterial flora. Bacterial flora refers to those bacteria that are normal residents of your body, particularly of your GI tract, but also with your skin. You have a very complex bacterial flora. That word flora is really kind of an artifact of the fact that bacteria used to be considered to be plants. Well, in the 1960s probably, a guy by the name of Whittaker proposed a 5-kingdom classification system. And these are the 5 kingdoms that are showing up here on this slide. Plants, fungi, animals, protista, and the monarans. Excuse me. What separates the monarans from these other 4 kingdoms in Whitaker's 5 classification system? Ariel? Prokaryotic and eukaryotic, right? Okay. Monarans are prokaryotic, and everything else is eukaryotic. This is a classification system that I grew up with. This is a classification system that we were teaching routinely until probably about 5 or 10 years ago. But things are changing. One of the things you will see about taxonomy is that taxonomic groups are constantly being revised. Particularly because the goal of taxonomy is to organize based upon evolutionary relationships. That's referred to as phylogeny. The early taxonomic schemes, particularly plant and animal, really did a very poor job of illustrating phylogenetic or evolutionary relationships among groups. Whitaker's system of 5 kingdoms did a much better job of driving that home. But still, there were some real limitations as to demonstrating true evolutionary relationships with this 5-kingdom classification system. You could have organisms that were in the same group that were very distantly related evolutionarily just because they had a similar appearance. A lot of this taxonomy was based upon physical characteristics and physical appearance. And those are not necessarily the best indicators of evolutionary relationship. Then came Carl Woese. And he kind of turned taxonomy on its head. And he developed a classification scheme that was not based primarily on physical characteristics, but that was based upon molecules, particularly RNA and DNA. Woese did a lot of work with sequencing of ribosomal RNA. And one of the things that he discovered was that the gene sequences that coded for ribosomal RNAs tended to be highly conserved, meaning they really didn't change much over time. Typically, when genes are highly conserved, they're really important genes that really can't tolerate a lot of mutation, or the organism won't survive. And ribosomal RNAs are critically important in making functioning ribosomes. And functioning ribosomes are essential to making proteins. So if we go back and start looking at ribosomal RNA genes, even minor modifications in the genes can really indicate very dramatic evolutionary splits on the family tree. So he started organizing organisms based upon their similarities or dissimilarities in their ribosomal RNA sequences. And when he did that, he found something quite interesting. What he found is that these 2 groups of bacteria down here in the monarans, called the bacteria and the archaea, were really very different from one another in their ribosomal RNA sequences. They were more different from one another than any of these organisms that were in the other 4 kingdoms. So what that told him was that evolutionarily, a diatom, or a amoeba, was actually more closely related evolutionarily to a human than this kind of bacteria was to this kind of bacteria. That was a mind blower. So Losey came up with a new system of classification that basically added another layer onto the classification scheme, the domain. He proposed a 3-domain classification system: the domain bacteria, the domain archaea, and the domain eukarya. The first two were both prokaryotic. The latter were all eukaryotes. So this contained 4 of the traditional 5 kingdoms, but what had been one unified kingdom in Whittaker's system was now split into these 2 domains. Basically what he was saying was, these are so fundamentally different from one another, as indicated by the ribosomal RNA sequences, that these diverged evolutionarily a long time ago and went their separate ways and evolved into their own groups of organisms. So when we talk about taxonomy in this course, we're going to use the 3-domain classification system. The newest classification schemes for bacteria, which we will talk about in a fair amount of detail, are all dependent upon this 3-domain system. So we're transitioning out of the 5-kingdom approach and now into this 3-domain idea of organization of living things. That's one of the misconceptions a lot of people have about science, is that science is a very static body of knowledge. It's not. Things are constantly changing in science. And one of the places where it really changes a lot is taxonomy. People are constantly splitting groups into smaller groups, lumping them together, getting new insights onto how organisms are related, and then the taxonomy schemes are revised to illustrate that new information or new strategy or new way of thinking. Okay. So let's talk a little bit about why we might study microorganisms. And let's break this down into 2 categories of why we might study things. We might be interested in studying microorganisms because they have beneficial activities. Also might be interested in studying microorganisms because they are harmful. So let's generate a little list of some beneficial activities of microorganisms and some detrimental activities. And then we'll dive into some of those in a little bit more detail to give you kind of a context. So, let's start out with bad.
They can make you sick.
Make you sick. There are a number of microbial diseases. What do we call a microorganism that causes disease?
Pathogen.
Pathogen. Good. So we'll spend a fair amount of time later in the semester talking about why are some microorganisms pathogenic and why are some not? What are some of the adaptations that they have that allow them to overcome the defenses of your body and cause disease? Your body has an incredible amount of defense mechanisms. And given the fact that microorganisms are everywhere, it's really a testament to the functioning of your immune system that we're not always sick. But sometimes microorganisms are able to gain the upper hand. Excuse me. So what adaptations do they have? What are some other detrimental activities of microorganisms besides pathogens?
Biodegradation.
Why would that be detrimental?
Obviously, one like anthrax bacteria, they release chemicals just to break down bodily tissues.
Okay.
It's not necessarily a disease in that case, it's more of a chemical reaction.
So maybe let's put this in the category of bioterrorism. Okay. Anthrax, botulism, all kinds of nasty toxins that microorganisms naturally produce. And if those could become weaponized or distributed into water supplies or food supplies, that's a real problem. That's a real concern. Other detrimental activities of microorganisms that we might be worried about. How many of you have refrigerators in your rooms? Pardon me?
Aging.
Yeah. Decomposition, spoilage of food. How much stuff do you have to throw out because it gets bad? Fair amount, right? Okay. Refrigeration will retard the growth of microorganisms, but eventually things will spoil, even in the refrigerator. We lose a tremendous amount of food worldwide to microbial decomposition. Some of that happens in the field. Some of that happens between processing and selling. Some of that happens after you sell things. And so one of the ways that we've addressed this is by loading foods up with preservatives that retard microbial growth. I think the last figure I saw was that the food preservative industry in this country is about a $30 billion a year industry. And if you go to the grocery store sometimes and to start to look at the listing of ingredients on package, a number of those, especially acidic compounds, are food preservatives. Typically, things that will dehydrate food will lengthen its shelf life. So salting, pickling, smoking, things like that. Making it more acidic. There are a lot of strategies that we'll talk about that we can use to deter microbial growth. Those are the three major detrimental activities that we'll talk about. And those are the ones that get the most popular press. What are some beneficial activities of microorganisms?
Bioremediation.
Okay. Y'all know what the term refers to? Using microorganisms to break down toxic substances in the environment. When we had the BP blowout in the Gulf, bioremediation was a big part of the strategy to deal with that. They used dispersants to try to break up oil slicks, but really the thing that really helped the most was introducing oil-eating bacteria into the water. And these were microorganisms that really metabolized the oil and used it as a food and ultimately converted it into carbon dioxide. Bioremediation can be used on toxic waste sites. It can be used on nuclear wastes. There are a whole set of strategies, particularly if we tweak genes of organisms and genetically modify them, we can oftentimes enhance their natural capabilities to break down substances. Some things are really even not easily broken down, even by microorganisms. PCBs, for example, are a man-made chemical that is very resistant to normal degradation. But we've been able to modify microorganisms to do a better job of breaking these things down. So that's a real benefit. What else? Nicole?
Can I ask a question about that?
Sure.
If you put these microbes in the ocean or whatnot, aren't they going to affect the ecosystem if they keep continuing growing?
That's a real concern, and some of them have been genetically modified so that they're kind of self-limiting.
Okay.
Yep, but it's still a concern, yeah. Absolutely. Anytime you do an introduction into an ecological system, you always have to be concerned about the law of unintended consequences. And humans have a very, very poor track record of trying to introduce organisms for what they think are a good reason into an environment. And then later they find out that there were consequences they hadn't considered. And sometimes the environmental impact is really severe. Other benefits. Why do we eat microorganisms? Yeah?
Like symbiotic relationships.
Sure. Agriculture is very dependent upon certain types of microorganisms. Y'all familiar with the concept of nitrogen fixation? Anyone up? Where is most of the nitrogen in our ecosystem?
In the air.
78% of the atmosphere is nitrogen gas. Can organisms use nitrogen gas to make nucleic acids and proteins? Very inert, very unreactive. But if nitrogen gas can be reduced to ammonium, for example, then that form of nitrogen is readily used by plants in their biosynthesis of nucleic acids and proteins. This is the process called nitrogen fixation. There are some natural ways in which nitrogen fixation occurs. Anybody know?
Probably do it with ultraviolet light, maybe. I don't know. It didn't really weird environment.
Lightning. Okay. In thunderstorms, some of the nitrogen is actually converted into ammonia. And then when it rains, that's washed down into the soil. But that's a very small amount. All of the significant nitrogen fixation is biological in nature. And it's carried out by bacteria as part of their metabolism. So we'll talk about this again when we talk about bacterial metabolism. Some microorganisms can do this in a non-symbiotic relationship. There are a group of microorganisms called the blue-green bacteria, or sometimes called blue-green algae, that can fix nitrogen by themselves. There's also a very important group of microorganisms that are symbiotic in nature with a group of plants. They live in the roots of the plants. They fix the nitrogen from the air and provide that nutrition to the plants. And the plants, through photosynthesis, produce carbohydrate, which the bacteria feed upon. What is the partner with bacteria? What group of plants are the symbiotic partners of these nitrogen fixers? Legumes? You heard of legumes before? Pea family? Clover, alfalfa, peas. Okay. If you pull these plants up, you'll see little nodules in their roots. And the nodules are where these nitrogen-fixing bacteria live. This is the concept behind the old agricultural practice of crop rotation, where you plant one year a corn crop, for example, which really tends to deplete the nitrogen in the soil. And then you follow that the next year with planting legumes, and they add nitrogen back to the soil again. So it's kind of a balancing act. So there are some definite agricultural benefits of microorganisms. What else?
The bacteria in our GI tract.
You bet. We have good bacteria in the GI tract. There are bad ones too. What do the good bacteria do for us? Why are they good?
Break down foods.
Okay, they could help in the digestion of some types of foods. That's not as huge of an issue for us, but it's very important in ruminants, for example. Yep, cellulose degradation. Okay.
I was going to say digestion.
Okay. There are some bacteria that through their normal metabolism produce vitamins. Vitamin K, for example, is produced by some of your gut bacteria. It's an important source of that particular nutrient. But the real big benefit is a phenomena called antagonism. Anybody know what that refers to? What happens if you go on long-term antibiotic therapy too sometimes? Yeah, you start getting other infections. For example, a yeast infection might pop up. Why did that yeast infection pop up? Why did taking an antibiotic cause a yeast infection? The antibiotics are killing the good bacteria. Are yeast susceptible to those antibiotics? They're eukaryotes. Most of the antibiotics work on prokaryotes. Okay? So what are these antagonists doing? Because of their growth, their metabolism, they're out-competing yeast cells for nutrients and things like that. They're suppressing their growth. They're keeping them down. They're limiting their populations. That can be true for yeast. It can be true for other pathogenic microorganisms. So if your normal flora gets out of whack, there can be some real health consequences. So what's a new therapy to restore your normal flora that's becoming kind of in vogue right now?
They put you on pills, usually they have bacteria sets in. Probiotics, yeah.
Probiotics are bacteria in pill form that you can take to try to restore your normal flora. Some people might think that this is kind of gross, but there's also been some research with what they call poop pills, where they actually take fecal transplants from people with a normal flora and transport that in pill form into people to try to restore their normal flora. So fecal transplants are kind of becoming, kind of the rage right now in terms of trying to treat particularly conditions like Crohn's disease, ulcerative colitis, that we think oftentimes originate when the normal flora gets out of balance. Okay? So yeah, good bacteria, real important to the functioning of your body.
You said transplant, like you're literally just making sure.
Yep. Other benefits. We haven't exhausted our list yet. Yeah. And we might as well throw antibiotics in there too. Inactivated forms of pathogens or their toxins can be introduced to give you immunological memory. So basically your immune system gets primed up by seeing a false threat, something that's been inactivated. And then if you see it for real, your immune system's all primed to respond quickly. It takes the immune system a little while to get geared up the first time you're exposed to something. The first time you're exposed to it is the pathogen. You might not build up a good immunity until you're pretty sick, maybe even dead. So doing this immunization with heat-killed bacteria or inactivated toxins like the diphtheria toxin, the pertussis toxin, the tetanus toxin, Tdap, okay, vaccination you've all had. Very important.
They use the heat to destroy the ribosomes, or
They use the heat to just really denature the proteins
okay.
so that they are still what we call antigenic. They'll still elicit immune response, but they're not active anymore. Their function's been changed. What about antibiotics? Where do antibiotics come from?
Lab accidents, penicillin.
Oh, okay, but what produced the antibiotic? They discovered it by a lab accident, yes.
Microorganisms?
Yep. Fungi and certain species of bacteria can both produce antibiotics. As a result of their metabolism, antibiotics are actually products of microbial fermentation. Any selective advantage for a microorganism producing an antibiotic?
Bacteria?
Possibly to suppress the growth of other bacteria. Basically conduct chemical warfare against your competitors. These two things have made probably the most impact on medicine of any discovery within the last couple of 100 years. What is a public health issue we are very concerned about in regard to antibiotics right now?
By killing all of the affected microorganisms, you're creating a less competitive environment for the organisms that aren't affected by it, and therefore you're creating resistant bacteria. Well, you're opening up room for resistant bacteria to grow.
The evolution of antibiotic resistance is a huge public health concern. And you all probably have seen articles at some point in time that said, can you imagine what it would be like to return to the pre-antibiotic era of medicine? That's very scary. Does anybody know when antibiotics really first came into widespread use? Hasn't been that long ago. Okay, they were really started being mass produced at the end of World War II. So really, the late 40s, early 50s is kind of the beginning of the antibiotic era. And in what, 65 years, we're now seeing a significant number of microorganisms that have evolved resistance to several antibiotics simultaneously. What's probably the poster child for antibiotic resistance? MRSA, okay, methicillin-resistant Staph aureus. We hear about that all the time. Other beneficial things that microorganisms do? You bet. Yeah, all the research that I mentioned before, absolutely. Photosynthesis. That planty thing. We tend to concentrate on green plants as being the primary contributors to the photosynthetic output. But in reality, about 50% of all photosynthesis is carried out by microorganisms. Many of them in marine and other aquatic environments. Photosynthesis not only gets us a carbon source for metabolism, it's also responsible for all of the oxygen in the atmosphere. Until the evolution of photosynthetic organisms, the atmosphere of the planet was a reducing atmosphere made up of a lot of ammonia, methane, CO2, but no free oxygen. So now we have to have oxygen to support the newly evolved strategy of cellular respiration, aerobic cellular respiration. What's another major ecological significance of producing oxygen to our planet? besides supporting aerobic respiration. The Earth is constantly bombarded by cosmic radiation, particularly output from the sun, like X-rays, gamma rays, and UV radiation. What filters a lot of that out?
Ozone.
Where does ozone come from? Oxygen? No oxygen, no ozone. So prior to the evolution of photosynthesis, could any organisms live on the surface of the land? No. Ultraviolet radiation and toasting. They could live in the water, though, because water does filter out UV radiation. But that transition of life from aquatic environments to terrestrial environments depended upon the formation of an ozone layer. That depended upon the evolution of oxygen by photosynthesis. Another big area of microbial importance is industry, particularly the food industry. There are all kinds of microbial foods, dairy products, some types of sausages, kimchi, pickling. Just a huge long list of microbial foods. We'll talk a little bit more about those some later on. And there are a number of chemicals that are produced by microbial metabolism. Various acids. In World War I, there was a Russian immigrant who came to England who figured out how to make acetone through microbial metabolism. Why was that significant? Because acetone was an ingredient that was used in the manufacture of gunpowder. And gunpowder was a limiting factor in World War I, having enough available gunpowder. So in some respects, microbial metabolism is credited with turning the tide of the war toward the Allies in World War I, because they figured out how to make more gunpowder. In some of the slides that are part of this PowerPoint that I post, and I post mainly illustrative slides, I don't really do much text. But if you look through there, there are a number of industrial products that are produced by microbial metabolism. And if you start looking through some of those, you'll go, wow, I didn't know that was a microbial byproduct. Wow, I didn't know that food was produced by microorganisms. Lots of benefits. Some downside. Okay. We're going to kind of refer back to some of these things a little bit more. So, the next thing we're going to talk about tomorrow is where microorganisms are found. And then we'll talk a little bit about the history of microbiology and get into some of the who's. Who studied microbiology, who made some of the more important contributions. I ran over, sorry, I didn't let you out early.